An elderly patient is rushed into the emergency department, presenting with altered mental status and sudden, violent generalized seizures. The clinical team immediately draws blood for a stat electrolyte panel. This telemetry panel flashes a critical panic value, a serum sodium concentration of exactly 110 millimoles per liter. At the bedside, the immediate clinical instinct is to hang an intravenous line of 3% hypertonic saline. The goal is to pull water out of the swollen brain tissue and halt the seizing. But as a clinical pathologist, your responsibility is to freeze the frame and question the data. If this number is accurate, the patient is experiencing a life-threatening osmotic crisis. But if this result is a pre-analytical collection error or an analytical artifact driven by circulating blood lipids, that aggressive saline infusion will destroy the patient's neurological architecture. To determine if this crisis is a true physiological emergency or a laboratory illusion, we must deconstruct this element down to its primordial physical chemistry. We start with the elemental identity. Sodium, derived from the Latin natrium, exists in human biological systems as a free, uncomplexed, monovalent cation. It functions as the chief osmotic anchor of the extracellular fluid. Because of its small ionic radius, paired with a single, dense, positive electrical charge, it exerts a powerful electrostatic pull on surrounding molecules. As you can see in this 3D model, a single sodium ion aggressively attracts the negative oxygen poles of surrounding water molecules, pulling them into a tight cluster. This rigid structure is called the radius of hydration. Notice how this heavy water shell physically prevents clean ion from passively diffusing through the hydrophobic lipid bilayer of a standard cell membrane. It is locked out. Because sodium cannot move freely, the human body uses it as a highly controlled physical tool. By actively trapping and manipulating this charged particle on one side of a membrane, the body dictates exactly where water must flow to balance the osmotic pressure. This volume control requires severe architectural compartmentalization. This volumetric bar chart illustrates the exact distribution of the body's total sodium mass. A quantified 95% of the functional sodium is sequestered in the extracellular matrix and the intravascular blood plasma, leaving a tiny fraction inside the cells. As we shift to this cellular cross-section, the contrast is stark. The extracellular fluid is packed with sodium at a concentration of up to 145 millimoles per liter, while the intracellular cytoplasm remains a near desert at roughly 10 millimoles per liter. To prevent those abundant extracellular ions from crashing through the membrane to reach thermodynamic equilibrium, every cell relies on an active transmembrane engine, the sodium-potassium ATPase pump. It continuously burns ATP to forcibly extrude 3 sodium ions out while pulling 2 potassium ions in. The metabolic cost is staggering. Maintaining this single gradient consumes up to 70% of a neuron's entire resting energy budget. By continuously ejecting positive charge, the cell builds an electrical potential energy floor. This stored kinetic energy powers secondary active transport, dragging essential nutrients like glucose and amino acids inside. Biological asymmetry is the foundation of life. If tissue hypoxia starves these pumps of ATP, the machinery halts. Sodium rushes in, dragging its heavy hydration shell with it, causing the cell to instantly swell and rupture. On a systemic level, defending that extracellular concentration falls to the renal architecture. Every single day, the human kidneys filter a load of 22,000 millimoles of sodium directly into the tubular lumen. To maintain blood pressure, the body must reclaim almost all of it. This flow map of the renal nephron details the specific extraction points. The proximal convoluted tubule reabsorbs 67% of the filtered load. The thick ascending loop of Henle pulls back another 25%. This leaves less than 1% to reach the final cortical collecting ducts. Here, the final volume is titrated by the renin-angiotensin-aldosterone system, mapped here in this connected cascade. A drop in renal perfusion triggers the release of renin, synthesizing angiotensin II, and culminating in the secretion of aldosterone from the adrenal cortex. Aldosterone binds to the principal cells in the collecting duct, upregulating epithelial sodium channels to extract the final remaining molecules of sodium from the urine and return them to the blood. This capacity varies by lifespan. Geriatric patients undergo a structural decline in renin and aldosterone production. Their natural safety margins compress, leaving them vulnerable to sudden fluid stressors. Terrestrial mammals evolved this hypersensitive conservation loop to fiercely hoard salt in ancient, salt-scarce environments. In a modern world of hyperabundance, this system continues to hoard every ingested molecule. This expands intravascular volume and increases cardiac output against stiffening vessels, transforming an ancient survival mechanism into a primary driver of chronic hypertension. When dietary intake outpaces water retention, the extracellular architecture collapses into hypernatremia. Defined clinically by a serum concentration exceeding 145 millimoles per liter, this state indicates a relative water deficit that concentrates the extracellular solute pool. This concentration spike generates a hyperosmolar force. As shown in this fluid dynamics simulation, the density of sodium outside the cell violently siphons water directly out of the intracellular space. The organic tissue shrivels. Inside the cranial vault, the cellular dehydration causes the brain mass to shrink. As it pulls away from the skull, the mechanical tension stretches and tears the delicate meningeal bridging veins. This physical rupture triggers focal intracranial hemorrhages, seizures, and neurological death. The opposite extreme is hypotonic hyponatremia. When serum sodium falls below 135 millimoles per liter with a parallel drop in plasma osmolality, the extracellular pool is diluted. This represents a primary failure in water handling, where excess free water is trapped in the blood. The hypotonicity reverses the osmotic gradient. This split-screen model contrasts the two extremes. While the shriveled cell on the left tears away from its vascular anchor, the cell on the right responds to the dilute blood by rapidly taking on water. It inflates against the rigid boundary of the skull. As astrocytes and neurons expand, the skull cannot accommodate the extra volume. Intracranial pressure spikes. The brain tissue is forced downward through the base of the skull, crushing the brainstem centers that control heart rate and respiration. Sodium imbalances weaponize osmosis to inflict lethal blunt force mechanical trauma from the inside out. If that hypotonic dilution develops slowly, over a period of more than 48 hours, the brain attempts a mechanical adaptation. To prevent swelling, the brain cells actively pump out their own internal organic solutes, molecules like glutamate and taurine, directly into the bloodstream. By reducing their internal osmolality to match the dilute blood outside, the osmotic gradient equalizes. Water stops rushing in, and the cells preserve their physical dimensions. This survival mechanism prevents acute herniation, but it creates a hidden clinical hazard. The brain has shrunk its internal capacity to survive the low-pressure environment. If a physician sees a critically low sodium value and infuses 3% hypertonic saline too quickly into this adapted state, the blood plasma suddenly becomes heavily concentrated. Those adapted brain cells are now defenseless against the steep gradient. The rapid concentration spike violently sucks the water right out of the adapted white matter. To prevent this catastrophe, medical guidelines enforce an unyielding mathematical speed limit. As detailed on this clinical blueprint overlay, a physician must never elevate the serum sodium concentration by more than 8 millimoles per liter within any 24-hour window. Violating this tiny kinetic margin causes the protective myelin sheath wrapping the central nerve fibers to strip away, fragment, and die. This structural destruction is called osmotic demyelination syndrome. It severs the descending motor pathways, leaving the patient with locked-in syndrome. Their cognitive mind remains conscious and alert, but they are trapped permanently inside a paralyzed body. Precision restraint is mandatory. Exercising that restraint requires verified certainty that the laboratory number is real. Many critical values are pre-analytical collection errors. Notice the phlebotomy draw in this visual. The needle is extracting blood from a vein directly downstream from an active IV line, infusing hypotonic maintenance fluid. This bedside error is called drip arm contamination. The IV fluid washes directly into the blood collection tube, creating a localized dilution illusion. The laboratory analyzer reads this highly diluted sample and generates a false hyponatremic panic value. If the clinical pathologist does not catch this pre-analytical signature by looking for a failed delta check, the clinical team might treat an artificial number.
Even perfectly drawn samples carry risks due to the automated architecture of high-throughput hospital core laboratories. Most automated chemistry platforms process thousands of samples a day using indirect ion-selective electrode potentiometry. The flaw of this method is phase volume displacement. This diagram illustrates a sample tube separating into its water and solid fractions. Indirect ISE algorithms assume plasma is 93% water. But when a patient has severe hypertriglyceridemia, lipid molecules physically displace the water. The mechanical pipette still draws its fixed total volume, but it scoots up more fat and less water than the algorithm expects. As we compare the two analytical methods, the indirect ISE applies a mathematical multiplier to a deficient water sample, spitting out a falsely low sodium reading, pseudo-hyponatremia. Next to it, a direct ISE cartridge analyzes undiluted whole blood. Because it measures the true water phase independently of the lipid volume, it captures the accurate physiological value. Trusting an automated analyzer without verifying the matrix physics can trigger unguided interventions based entirely on ghost numbers. To avoid these traps, modern pathology enforces diagnostic stewardship. Repeating serial electrolyte panels within a single day on stable wards is restricted. Because fluid shifts take extended periods to safely equilibrate, rapid repetitive draws only yield redundant data and drive unnecessary costs. Pathologists also utilize mathematical correction formulas for translational shifts. In acute diabetic ketoacidosis, massive blood glucose concentrations shift water out of the cells, diluting the measured sodium. This requires a mathematical correction of the value, not clinical fluid intervention. The diagnostic axioms are clear. Guard against the lipid displacement mirage, respect the strict 8 millimole demyelination speed limit, and always track sudden drops for IV line contamination footprints. This holographic blueprint displays the foundational framework. Sodium operates as the primary extracellular osmotic anchor, the potential energy floor for secondary transport, the regulator of cell volume, the variable driving the RAS endocrine feedback loops, and the potentiometric target for laboratory quantification. Clinical mastery requires tracking this element's physics from the lipid envelope of a brain cell straight through to the glass capillary membrane of a laboratory analyzer. Respecting this primordial element's power is the key to managing human survival.